This is part 2 of the walkthrough of the AQA GCSE Biology 2022 Higher Tier Paper 1. Okay, question 6. It tells us, Table 6 shows us information about 5 different organisms. So it's given us A, B, C, D, E, and then it's given us the surface area and the volume for all of them. But the surface area to volume ratio for organism C is missing. Calculate value X in table 6. Give your answer to the nearest whole number, 3 marks. So we need to find the surface area to volume ratio for C. What we'll need to do is we'll need to take the surface area, which for C is 9.96 times 10 to the power minus 3. And then we need to divide that by the volume, which is 1.35 times 10 to the power minus 4. And then plug that into our calculator, and then we should come out with a value of 73.7 recurring. As it says, the nearest whole number will be 74. 6.2, what is the relationship between the size of an organism and its surface area to volume ratio? Use table 6. Okay, so going back, what can we notice from our table? We can see that the surface area to volume ratio for each of these organisms from A to E is getting smaller. Even if you plug in R1 now, you can definitely see that. So what's happening to the surface area? The surface area seems to be getting larger. So what does that tell us about the size of an organism compared to surface area to volume ratio? The larger the organism, the smaller the surface area to volume ratio. 6.3. Organism B exchanges gases with the environment directly through its skin. Organism D exchanges gases with the environment using its respiratory system. Explain why organism D requires a respiratory system, but organism B does not require a respiratory system. So when it's mentioning gases with the environment, we need to think about the process in which these gases will move. So the process we should be thinking about is diffusion. But why would organism D require a respiratory system to carry out diffusion, whereas organism B can just do this through its skin? So let's look at our table again and see how organism B and D are different. So what do we notice between B and D? Organism B has a larger surface area to volume ratio. Organism D has a smaller surface area to volume ratio. So how does that link to the process of diffusion? Well, let's clarify with our first point. Organism D has a smaller surface area to volume ratio. And again, we need to think, how is that linked to diffusion? So if the organism's gases that it wants to intake are in the environment, and we want that to get into the organism, if the surface area to volume ratio is smaller, that will mean that the diffusion will take longer. The other thing that could be said is the diffusion pathway is too large. Table 6 is repeated below, so the table is shown again. Table 7 shows information about organisms D and E. So metabolic rate in arbitrary units for organism D is much higher than it is for E. So if we analyze the table now, organism D has a larger surface area to volume ratio than organism E. Yeah, its metabolic rate is much higher. So there must be some link there. Let's read on. organisms D and E both keep a constant body temperature, means they're warm-blooded. Explain why the metabolic rate of organism D is greater than the metabolic rate of organism E. Use information from Table 6 and Table 7. So we analyzed the table and we saw that organism D has a larger surface area to volume ratio. So we should be stating that once again. So organism D has a larger surface area to volume ratio. But how now is that going to be linked to constant body temperature? Well, let's think about this metabolic rate. This is the rate of chemical reactions occurring in the body. But what might be the link between metabolic rate or metabolism and body temperature? Well, if we think about an example of a metabolic process, respiration, this is supposed to be an exothermic reaction. So that means that heat energy is given to the surroundings. It leaves our organisms. So if we have a larger surface area to volume ratio, that will then mean that the organism D will then lose more heat quickly. So if it's going to lose more heat quickly, then it needs to keep that constant body temperature. So that would then mean that the rate of respiration will need to be higher in organism D. Let's make sure that we make it clear that respiration is an example of a metabolic reaction. So that will then mean that it will need to keep carrying out this respiration to keep itself warm by producing more heat. (6.5) Organism D and organism E both have alveoli in the lungs and villi in the small intestine. Figure 8 shows some alveoli and some villi. So we've got alveolus here and we've got some villi here. What should we know about these two structures? Their functions we need to know, and we need to know how they are adapted to carry out their functions. Describe how the alveoli and the villi are adapted to increase absorption. So we need to talk about how both of these structures have changed in order to increase absorption. Absorption is that funny word, it's a bit vague, so we need to think about what processes will these structures carry out. So we should be thinking diffusion again. So this is related to the exchange of substances across surfaces. So if we analyze the pictures and use our knowledge, they have some things in common. So what do both of these structures have in common? Well, they both cause an increase in the surface area. Now, why does that do that? Why is that important? So that diffusion is maximized. Okay, let's think also. We see these cells here. What is special about these cells? Well, these cells that line our villi and that line the alveoli, they're only one cell thick. So the walls are one cell thick. So both have walls one cell thick. So what does that do? So the diffusion pathway is shorter. So diffusion pathway or diffusion distance is shorter. And of course, that will mean then that also the rate of diffusion will be higher. Anything else that we can spot? Well, what's surrounding our alveoli and also is very close to our villi, these are supposed to be capillaries. So we can mention something about the capillaries now and how that will help. So the thing that we can say is both have a good blood supply, and that also means that they have a network of capillaries surrounding them or next to them. What does this do? This maintains the concentration gradient. So there's always a steeper concentration gradient, so that means diffusion will continuously occur. Maybe we can start to think about individually how these might be different. Or how are the villi a bit different, how they adapted further? They have microvilli. And that again, similar to the villi and the alveoli, which further increase the surface area. And what does that mean? That means diffusion, again, will be maximized. Question 7. Human immunodeficiency virus, HIV, is a pathogen. Give one way HIV can spread from one person to another person. Well, remember, we only need to give one way. Just make sure that we don't give more than one, or else we could prevent ourselves from getting a mark. So HIV, one main way it can spread is through sexual contact. But if we had to think of another way, just to make sure that we are good, or if the question was two ways, the other way would be through bodily fluids. Table 8 shows information about new cases of HIV diagnosed in the UK. Describe the trends shown in Table 8 between 2010 and 2018. So we're looking at two cases changed over a period of time, one for women and one for men. So we could start to look at how women changes only, and then separately we could see how men changes only, or we could talk about how they overall change for both of them. But if we do that, that's probably going to be just one mark. So it would be safer to describe how women changes, get a mark, and then how men changes and get a mark. So let's look carefully at the data, because what they can sometimes do is put some numbers going up and down, also known as fluctuating, just to catch students out. So let's look individually at how these numbers change. So 376 decreases to 361. Then it increases to 397. Then it decreases to 298. And then it decreases further to 242. Okay, so we can see that there is a decrease, then an increase, and then further decreases. Let's look at how the men changes from 2010 to 2018. There is an increase, and there is another increase, then there is a decrease, then there is a decrease. So if we want to put this into words now, for women, the cases decrease, then increase, and decrease, which is fluctuating. For men, the cases increase, and then decrease. 7.3. Suggest one reason for the change in the number of new HIV cases between 2014 and 2018. So looking at the table now, we just have to focus on these sections of our table. So what do we notice? They are both decreasing between those years. So why would the number of cases decrease for HIV? Well, there could be better forms of treatment. For example, drugs. Remember that one of the ways to treat HIV will be with antiretroviral drugs. If we needed to give another reason why, because science is developing, there is better understanding or education about HIV. We could mention the other forms of prevention, which are condoms. However, what we would need to be careful of is the fact that at 2010, there were condoms being used. At 2012, there were condoms being used. However, what might cause the decrease, as we see between these years, it may be the fact that the condoms are more accessible, widely available, or cheaper, which would mean that more people would be using them. 7.4. Calculate the ratio of new HIV cases in women to new cases of HIV in men in 2018. Give your answer to 3 significant figures. So going back into our table, looking at 2018, and we need to take the women value 242 and the men value 1288. So our women to men will be 242 to 1288. Now if you look carefully at our answer here, we need to have a value to 1. So our men value needs to be 1. So that would mean that we would need to divide both sides by 1288. So we know now that for men that would be 1. So plugging that into our calculator, we should come out with a value of 0.1878. Keeps going. However, as it says 3 significant figures, we can only have 3 values after our decimal point. So we'll have 1, we'll have 8. However, we need to round it because it's greater than 4. So then it would be 0.188. 7.5. In the UK population, the total number of women is greater than the total number of men. The data in Table 8 is used to compare the proportions of new cases of HIV in the population for men and women. Suggest how the data could be presented differently so a more valid comparison can be made. So, finally going back into this table, so they've told us that the population of women is greater than the population of men. So that will mean that the data is going to be a bit skewed now. So we need to think about how could we better show this data to allow comparisons between each of those cases in those different years. So the easiest way, which has probably been seen in exam questions so many times, is by using percentages. Because what you would then do is you would take that value and then put that as a percentage over the total population, wherever that number is, multiply it by 100. The other way that this could be done is by comparing that to a certain number of people. So for example, you could compare that number in the table to 100,000 or 500,000, 10,000, any number which can represent a large proportion. Question 7.6. Scientists have been working to produce a vaccine for HIV for many years. Explain how a vaccine for HIV could work to prevent a person developing HIV infection. So we need to describe what this vaccine is and then how that's going to prevent further infection. So we've got to really now think about this graph of our immune response. If we are able to explain this graph carefully, we should be on our way to 4 marks. So first thing that we need to think about is what would happen with a person that is going to take this vaccine. So thinking of what the virus is, a person is injected with a weak or dead version of HIV. So once that HIV virus portion is injected into the body, what will the body do in response? So we don't want to write the word body, we want to write the specific part of our body which will respond. And that will be the white blood cells, which will produce antibodies against HIV virus. How do antibodies work? Well, they bind to the HIV virus and destroy it. So what we've done here is we've described this first portion of our graph here. The reason why the line increases is because the antibodies have been produced by the white blood cells. Why have they been produced? Because there's been an injection of a virus at the first stage of the graph. The reason why the line goes down is because the antibodies have done their job. They've destroyed the virus, so they don't need to be produced as much anymore. But we need to think about the next part of our graph, which is this section where our line starts to increase. So just to be clear, this line is supposed to represent antibody concentration, and this is over a period of time. So what is the reason why there is such a rapid and large increase in the concentration of our antibodies? Well, it's because our white blood cells have somehow remembered this specific virus shape and is going to quickly produce antibodies against it. So we want to ensure that we write, if infected again, antibodies will be produced quickly. And some of you may have been taught that this is due to the presence of memory cells. But as this is not part of the specification, this is not really going to be worth a mark. What we must say is if we are infected again with this virus, the antibodies will be produced, which are specific to HIV. They will be produced very quickly. A person with late-stage HIV infection has AIDS. Scientists have produced monoclonal antibodies for HIV. The monoclonal antibodies can prevent a person infected with HIV developing AIDS. Describe how the monoclonal antibody for HIV can be produced. 4 marks. So we have to right here recall the process of producing monoclonal antibodies. So first thing that must be done. Now remember, they are mentioning HIV again. So we need to, in our answer, be specific to the virus HIV. So first step that must happen in the process of producing monoclonal antibodies is there needs to be an injection of this virus into a mouse. So injecting HIV virus into a mouse. What will happen then? Now, similar to our vaccine, there will be some production of antibodies, absolutely. However, in this case, we now need to specify the type of white blood cell which is producing antibodies. And this is called the lymphocyte. So lymphocytes are produced against HIV, which means that they will produce antibodies which are specific to HIV. What do we do with these lymphocytes? The lymphocytes are collected and we fuse these lymphocytes together with a tumor cell. What is that combination of tumor cells and lymphocytes called? Hybridomas. Once these hybridomas have been created, we then want to clone these hybridomas. Why do we want to clone them? So we have more of the specific antibody against the HIV being produced. So when you're thinking about the process of producing monoclonal antibodies, the key words that need to be mentioned are lymphocytes, hybridomas, and then after that, they're going to be cloned. Question 7.8. Figure 9 shows how HIV enters a human cell. So parts of it are labeled, especially the things that we want to focus on: HIV antigen binding site and a HIV genetic material. Suggest how the monoclonal antibody for HIV helps to prevent a person infected with HIV developing AIDS. Use information from figure 9. So we've got to analyze this figure now and then try and formulate an answer. So we have a human cell which has got on its surface a HIV antigen binding site. We have a HIV virus with a HIV antigen. So what should happen then is the HIV antigen should just bind to the binding site as it's shown in the second section here. Once that's happened, our HIV virus is now fusing with our human cell, and the genetic material is then entering the cell towards the nucleus. So we need to think how having a monoclonal antibody is going to prevent this from happening. So our monoclonal antibody should have a sort of shape such as this. So maybe what will happen, because remember that this is a suggest question, so we need to think logically what is the best thing that is going to happen here. The monoclonal antibody is probably specific to this HIV antigen. What does that mean if it is specific? It means then that it will bind. What did we say that antibodies can do? Antibodies can help to destroy pathogens. But if we're using the figure, if our monoclonal antibody binds to our HIV antigen, that then means that the HIV cannot bind to our human cells. And then what does that then mean? It means that it can't release the genetic material to our cells.